Hello, today we are going to study about phylum Protozoa of kingdom Protista. So what are protozoans? Basically these are microscopic acellular यूकैरियोटिक एनिमैक्यूल्स. ठीक है, या तो सॉलिटरी दैट मीन्स अकेले पाए जाते हैं या कॉलोनीज मतलब एक ग्रुप में पाए जाते हैं. ठीक है, बेसिकली इन व्हाट मीडियम, एक्वेटिक मीडियम, or as parasites that is on other living organisms. So the first time it was discovered by A.V. Leuwenhoek. Okay, the year is also mentioned here, that is 1672, and the term protozoa was coined by Goldfuss, that is 1809. Okay, so here are the general characters of ये phylum protozoa. Basically, firstly we will discuss the body forms. So the body forms are highly variable because sometimes they are irregular like in amoeba. Okay, sometimes they appear in as spindle shape in euglena. Okay, slipper like in paramecium, as we all know, and bell like, that is we have seen the temple bells, so bell like in vorticella. Now body organization, these are acellular. Acellular means they have simple cell organization. Okay, so they cannot form tissues and organs, and that is इनका जो ग्रेड होता है वो प्रोटोप्लाज्मिक होता है. तो प्रोटोप्लाज्मिक मतलब साइटोप्लाज्म प्लस न्यूक्लियोप्लाज्म. ओके, नाउ द size is microscopic, of course, obviously, and the color wise they are colorless. Okay, the next thing is symmetry. Okay, they can be asymmetrical, that is irregular symmetry. Okay, so they cannot be symmetrical in any way, that is paramecium and amoeba, and they can be spherically symmetrical, that is volvox. They can be radially symmetrical, that is euglena, or bilaterally symmetrical, that is giardia. Now the next thing is body structure. Okay, what kind of body structure they have? So they are typical eukaryotes. Okay, so they show division at the level of cell organelles. As I have mentioned before, that they do not form organs and tissue, but they have cell organelles. So they can be uninucleate, that is having a single nucleus. So the example is amoeba. Okay, and they can have two nucleus, that is binucleate. So the example is giardia, and all ciliates, that is having the organism which are having cilia, and they can be multinucleate, that is Plasmodium. Okay, the examples are mentioned here. Okay, the nuclei can be monomorphic. Okay, then the type of nuclei. Mono means single, morphic मतलब form, morph. Okay, in एंड इनर एंडोप्लाज्मिक रेटिकुलम मतलब कि अंदर और अंदर वाले जो एंडोप्लाज्मिक रेटिकुलम के अंदर भी ये प्रेजेंट होता है. इसकी अलग अलग फॉर्म में भी प्रेजेंट हो सकता है. ये नेकेड भी हो सकता है मतलब विदाउट एनी कवरिंग. ओके, पेल्यूकुलेटेड मतलब पेलिकल की कवरिंग होती है, दैट इज पैरामीशियम में पाई जाती है, या फिर उसकी शेल्ड कवरिंग होगी. ओके, एक शेल होता है जैसे स्नेल्स में आपने देखा होगा, वैसे ही इसमें शेल की कवरिंग होती है. तो शेल की कवरिंग में एफिडियम आ जाता है एज एन एग्जाम्पल. ओके, और कुछ जैसे इंटरनल सपोर्टिंग एलिमेंट्स भी पाए जाते हैं इसमें, जैसे कि एक्सोस्टाइल. वो उसका एग्जाम्पल है ट्राइकोमोनास. एक्सोस्टाइल भी एक टाइप की पेल्विक रीजन में पाई जाती है, जो स्टाइल एक इसकी ऑर्गेन टाइप होता है. ओके, एक्सियल फिलामेंट. अब फिलामेंटस फॉर्म भी पाई जाती है. एक्सियल मतलब कि एक्सिस पे लोकेटेड है, दैट इज द टॉप ऑफ द बॉडी पार्ट. ठीक है, और ये एक्सोपोडिया में लोकेटेड होता है ऑफ एक्टिनोफायरस. ठीक है, और इंटरनल शेल्स भी प्रेजेंट होते हैं. ठीक है, जैसे कि एकेंथोमेट्रा में. नाउ द नेक्स्ट कैरेक्टरिस्टिक इज ऑर्गेनेलीज ऑफ लोकोमोशन. कौन से जो ऑर्गन्स होते हैं वो उनकी मूवमेंट में हेल्प करते हैं फ्रॉम वन प्लेस टू अनदर. तो ये टेम्परेरी भी हो सकते हैं. ठीक है, फिंगर लाइक फॉल्स फीट जो जिन्हें सूडोपोडिया कहा जाता है, ये अमीबा में प्रेजेंट होते हैं. या फिर लॉन्ग थ्रेड लाइक, जैसे कि फ्लैजेला हो गए. छोटे छोटे सीलिया हो गए जैसे कि पैरामीशियम में प्रेजेंट होते हैं, या एब्सेंट ही होंगे कुछ पैरासिटिक फॉर्म्स में जैसे कि स्पोरोजोअन्स. मैक्सिमम जो पैरासिटिक फॉर्म्स होते हैं वो मिनिमलाइज करने की कोशिश करती हैं ताकि वो होस्ट पे ज्यादा टाइम तक सर्वाइव कर सके. तो वो कुछ ऑर्गन्स जो होते हैं वो गिव अप कर देते हैं. तो जैसे कि एब्सेंट होता है स्पोरोजोअन्स में. दिस कैरेक्टर फॉर्म्स द बेसिस ऑफ क्लासिफिकेशन ऑफ प्रोटोजोन. तो जो प्रोटोजोन होते हैं एक्चुअली वो ऑर्गन्स ऑफ लोकोमोशन पे ही क्लासिफाई किए जाते हैं मोस्टली. नेक्स्ट थिंग इज न्यूट्रिशन. तो प्रोटोजोन जो होता है वो मोड ऑफ न्यूट्रिशन में भी वेरी करते हैं, जैसे कि होलोजाइक या ऑटोट्रॉफिक हो गए, ठीक है, मतलब वो अपना खाना खुद प्रिपेयर करते हैं, जैसे कि यूग्लिना, वॉल्वॉक्स, ओके। एंड वॉल्वॉक्स तो जो होता है वो कॉलोनीज भी बनाता है, ओके। नेक्स्ट इज होलोजोइक मीन्स एनिमल लाइक, जैसे कि पैरामीशियम, ओके। तो होलोफाइटिक या एट्रॉपिक, ये दोनों सेम ही टर्मिनोलॉजीज है। अगर ये आ जाए तो एग्जाम में अगर मेंशन्ड हो तो कंफ्यूज नहीं होना होता है। ऑटोट्रॉफिक का वही मतलब होता है जो होलोफाइटिक का होता है, ओके। होलो मीन्स एनिमल लाइक, फाइटिक मीन्स प्लांट, तो होलोफाइटिक। होलोजोइक मीन्स completely animal like, okay, as in paramecium. So saprophytic, next is saprophytic. So saprophytic as in trypanosoma. Here, trichonympha is also mentioned as an example, and the last form is parasitic form, like in Plasmodium. प्लाज्मोडियम एक पैरासाइट होता है जो मस्कीटो में पाया जाता है, ओके। तो द सेम parasitic form is Plasmodium. Holozoic forms have a definite cytostome. Mouth, here I have mentioned that stome जब भी सफिक्स में आ जाएगा तो उसका मतलब माउथ होता है, एंड फाइट जब भी आ जाएगा उसका मतलब एनस होता है। सो साइटो मीन्स साइटोप्लाज्मिक, सो फाइट मीन्स एनस, साइटो मीन्स again the cytoplasmic thing, that is cell, and stome means mouth. Next is respiration and excretion. They occur through general body surface, so they do not exhibit any organs or tissues. So generally the respiration and excretion occur through body surface only, okay, because they are basic cell aggregates. In excretion they are generally amonotelic, so remember this thing, that they are amonotelic. Now next thing is osmoregulation. Freshwater protozoans like euglena, amoeba and paramecium etc have one or more contractile vacuoles, that is they can have more than one contractile vacuole for osmoregulation, which is analogous to kidney in vertebrates, okay. So this is an analogous organ, um, analogous to what, kidneys in vertebrates. Contractile vacuoles are absent in marine, so okay, so they are present in freshwater but they are absent in marine forms like Eliphidium, and parasitic forms like Entamoeba forms due to their isotonic nature, because they have a isotonic nature, so maintain the water balance, these contractile vacuoles are absent. The last but not the least characteristic is reproduction. All the protozoans show asexual reproduction, which may be binary fission, okay, or multiple fission. So the examples are given. For multiple fission, amoeba is there and plasmodium, and for the binary fission, euglena and amoeba. Amoeba shows both multiple fissions also and binary fission also. Okay, so it is this is the obligatory thing in this, or budding or sporulation. So when it is mentioned budding or sporulation, budding means developing of the same individual from the same individual, okay, by certain separation, and sporulation means formation of numerous spores. So, okay, okay. So some protozoans, especially ciliates and sporozoans, show sexual reproduction. Example conjugation and automogamy. Example in Paramecium. And sporozoans generally show alternation of generation, that is Plasmodium. So they show alternation of generation in the same lifespan. Encystment. Okay, so what do you mean by encystment? Encystment means formation of protective covering, that is a cyst-like covering to protect itself from unfavorable conditions. So all freshwater organisms like amoeba and euglena, and many parasitic forms like entamoeba, histolytica and giardia form this thing called encystment. The process of forming cyst is called encystment. So it also helps in dispersion, and the main thing here is that it only forms in freshwater species, not in marine forms, okay. Now next is habitat. Habitat greatly varies, okay. So they can be freshwater habitat, marine, symbiotic, commensal. Commensal means the negative thing, negative relationship, okay. The freshwater example is amoeba, paramecium. Marine is Aphelidium. Again, parasitic is Entamoeba histolytica and Trichomonas, Trypanosoma, and symbiotic example is Trichonympha. Commensal is Opale lineata. Now let's move to the unique features of phylum Protozoa. These are acellular organisms with protoplasmic organization, that is again cytoplasm plus nucleoplasm. Division of flavor is at the level of cell organelles, and Organelles of locomotion may be pseudopodia, example amoeba, and flagella, example euglena, and cilia, example paramecium, or absent. Okay, the nutrition mode of nutrition may be holozoic, saprophytic, parasitic, or only a few forms are autotrophic. Most of these are heterotrophs. Presence of one or more contractile vacuole, as I've told earlier, there may be presence for osmoregulation in fresh water. Okay, in marine forms it is absent. So these undergo the asexual reproduction by binary fission or multiple fission, and both by amoeba. Some undergo sexual reproduction, that is conjugation, autogamy, and endomixis. All freshwater and many parasitic protozoans undergo encystment for dispersal of and pollination. Next is classification. So protozoans are grouped along with slimoles. So remember that slimoles is a separate kingdom. Okay, but along with it, it is also classified. So Protista algae, including they include photosynthetic protists, that is, they can perform photosynthesis like plants, and Protestant protozoa include heterotrophic protists, that is, they depend on photosynthetic protists. Next is classification. So they are subdivided into subphylum, that is Sarcomastigophora, Sporozoa, Nidospora, and Coeliophora. So let's discuss this. So the organelles of locomotion, on the basis of organelles, they are also divided. First, in Sarcomastigophora, you will find pseudopodia or flagella or both. The Sporozoa and Nidospora lack these, so they are absent. And Coeliophora, as the name tells us, the cilia is present. Next is nucleus. So nucleus can be monomorphic, that is single morphism, and in all the three, monomorphic nucleus is present, but in Coeliophora, dimorphic nucleus is present. Next is habitat. They can be free living or parasitic in Sarcomastigophora, and Sporozoa and Nidospora are all parasites. In Coeliophora are free living or they can be parasitic also. And the reproduction part absent or syngamy. They also show syngamy, and Coeliophora show conjugation. Next is spore formation. The spores are not formed here. In this state, sporozoa, infective spores are formed. Okay, and Nidospora spores have one or more polar filaments and one or more sporoplasms. So spores plus cytoplasm forms sporoplasm. Next is Coeliophora. So Coeliophora, no spores are formed. Okay, and daughter cells are formed by transfer or binary fission. Next is subphylum. Now let's discuss the Sarcomastigophora in detail by dividing into subphylum, that is Sarcomastigophora. So it has three superclasses, that is Mastigophora, Opalinita, and Sarcodina. So they are divided on the basis of locomotion, body covering, nucleus, and mode of binary fission. Here are the three superclasses mentioned of Sarcomastigophora, that is on the basis of organelles again and characteristic. So Mastigophora, one or more flagella, they have presence of one or more flagella. In Opalinta, there are cilia-like, that is, their whole body is covered with cilia, and Sarcodina have pseudopodia. Now, next is body covering. So, the body covering in these two, it is generally pellicle. In Sarcodina, it is generally plasma membrane, and some are shelled, that is, shell-like covering. Okay. The next is nucleus. So, generally one or two, like in Gaillardia, for example, it is found two or more monomorphic nuclei found in Opalinata and in Sarcodina, generally one but many in Pelomycia. So it differs from subphylum to subphylum. And the last is binary fission. So it is longitudinal in Mastigophora, plasmotomy, that is involving the plasma in Opalinata, and in Sarcodina it is irregular. Now the superclass Mastigophora is further divided into two subclasses, that is Phytomastigophora and Zoomastigophora. So what are these? Phyto means plant and zoo means animals. So like the name suggests, it is on the basis of the chloromatophores present. So there is presence of chloromatophores, so they are plant-like. The chloromatophores are absent in Zoomastigophora, so they are animal-like flagellates. So it is absent in these animal-like flagellates. Now let's discuss the nutrition. So as we know that in plants, autotrophic nutrition is found, and reserve food material is starch or paramecium or oil. Okay, and in heterotrophic and heterotrophic, the reserve food material is mainly glycogen. Now next is number of flagella. So there is one flagella in Euglena and two in Cryptomonas. Here there is one flagella in Trypanosoma and more flagella like 8 in Giardia and many in Trichonympha. Next is habitat. So these have aquatic habitat, that is freshwater or marine. They can be both or either of these two, but they are mostly endoparasitic and pathogenic. So there's also the nutrition. The mode of nutrition is also endoparasitic and pathogenic, and they are symbiotic. So the mention here is mentioned the examples, that is Trypanosoma, Giardia, and symbiotic in case of Trichonympha. The examples is Amoeba, Cristomonas, Ecligna, Volvox, Gonistomium, and Noctilium. And the example here is Giardia, Trichonympha, and Trichomonas. एक ही याद रखो. No need to remember all these. Next is superclass Opulenta. It is characterized by endochemensals. Okay. So found in rectum of anurans. What are anurans? A group of amphibians that include frogs and toads, characterized by absence of tail in the adult stage. Okay. So tailless amphibians like frogs and toads. Body is covered by fork pellicle. Organelles of locomotion are mainly small size. Cilia-like flagella present in oblique rows over the entire body. So they are present in the entire body surface, and two or many monomorphic nuclei are also present. Cytostome is absent and shows saprozoic nutrition. So asexual reproduction by pelliculous somatogenic band diffusion, also called plasmotomy. So here you need to remember the key word, that is plasmotomy. That is cytokinesis precedes karyokinesis. Cytokinesis, cytoplasmic division, and karyokinesis, nuclear division. And the next is sexual reproduction, that is occurs by syngamy, fusion of flagella and isogametes. And iso means different. Okay. Example Opilina and Zereraria. Now let's discuss the superclass Sarcodina. So superclass is divided into two classes, Rhizopodia and Actinopodia, on the basis of type of pseudopodia and mode of living. Now here let's discuss the two classes of superclass Sarcodina, that is Rhizopodia and Actinopodia. They are classified on the basis of type of pseudopodia. So that is in Rhizopodia, Lobopodia is present, or it is also called Philopodia or Reticulopodia depending upon the structure. If it is filiform, that is Philopodia, it is has network, so it is called as Reticulopodia. As we all know, the term for network is reticula. Okay, and it is lobiferous so Lobopodia. Lobo means the type, the structure, and podia means feet. Exopodia means axial podia, axial feet. Okay, then the mode of living is creeping type. So they are creeping, that is, they can only creep and crawl on the surface. And in Actinopodia, they are sessile or floating type. Sessile means they cannot move. Okay, the another terminology for this is sedentary. Example Amoeba, Entamoeba, Diffuga, Polystomela, and Govidia. Next, for actinomorph, the mostly example you can remember that Actinopodia, so Actinophyrus and Acanthometra. Also, Zoo Chlorella is also the example. Now, the subphyla of Sporozoa. So, subphyla of Sporozoa are classified into three types: Telopodia, Toxoplasmia, and Haplopodia on the basis of nature of spores and reproduction. So, there are three classes of subphylum Sporozoa, that is Telopodia. Now, they are characterized on the basis of nature of spores, mode of reproduction, and the examples of spores. So, spores are without polar capsules and polar filaments in Telopodia. The spores are absent. Spores are present in some cases, okay, in case of haplosporia. Next is mode of reproduction. So here both sexual and asexual reproduction happen in the life cycle. Here only binary fusion happens. Okay, that is asexual, and here only schizogony happens. That is asexual. So examples is Gregaria, Monocystis, Emeria, and Plasmodium. Here the example of toxoplasma, as you can learn this from the title itself, that is toxoplasm. Here haplospores, so Coriosporidium. Next is subphylum Nidospora. So it is divided into two subclasses, Myxosporidia, Microsporidia, and on the basis of nature of spores, origin of spores, and host type. So there are two subclasses, Myxosporidia and Microsporidia. So they are classified on the basis of nature of spores, origin, host type, and example. With two or three valves, the nature of spores, okay, the spores are present with two or three valves. Here only one valve is present. They are multicellular in origin and they are micro are unicellular in origin. Now the host type is cytozoic parasites in fishes. They are present, okay, and cytozoic parasites of arthropods and vertebrates are present. They are talking about the phylum Arthropoda. Now, example is Myxobolus. As you can learn this from the title itself, Myxobolus, Ceratomyxia, and Myxedium. Here, they only have one example, that is Nosemia. So, Nosemia is a genus of microsporidium by infecting insects and other vertebrates, often affecting the honeybees. Subphylum of Celiopodia. As the name suggests, the subphylum of Celiopodia includes Ciliata, which is further divided into four subclasses: Holotricha, Peritricha, Subtoria, and Spirotricha on the basis of body ciliature and buccal ciliatures. Buccals mean the cilia which are present at the mouth region. Okay, they are further classified on the basis of body cilia and buccal cilia and examples. So the holotricha, the cilia is uniformly distributed in the body. In peritricha, no cilia in the body of in adult in sedentary forms. Okay, so it is not present in the adult of the sedentary. Sedentary means they are sessile, they cannot move. Suctoria, adult with no cilia with suctorial tentacles and are sedentary. Okay, and here Spirotricha, they are reduced. In buccal ciliatures, absent, they are absent or inconspicuous. That is, they are present but they are not visible. The term inconspicuous means that. Okay, in Peritricha, they are anti-clockwise of या फिर strong उसमें present होंगे, and here they are absent, and in Spirotricha they are well developed. The buccal ciliatures are well developed. Examples is Denium, Balanotidium, and Vorticella, Podophyra, and Ephelolata. Here the example is Nyctotheras, Stylonosia, and Saprodinium, of course. Now, this is the outline classification of phylum Protozoa. So you can just overview this, and next we move to the next topic, that is locomotion in protozoa. So locomotion is basically defined as the movement from one place to another, okay, either in liquid or gaseous medium. So mainly in biosphere we use the air as a medium. So locomotion like creeping, walking, running, hopping, flying, etc. It is found in most except sponges because they are sedentary, sessile, they cannot move, and silent, and most of the parasitic invertebrates. So you can just get go through the glossary to get the familiar with some of the terminologies mentioned here. Now next is significance of locomotion. So what is the significance of locomotion? It gives protection from the predators. Okay, they that means they can move, so they can easily protect from this, to search and to search for food and water, to transfer animals from unfavorable to favorable environment, that is migration, and to mate for reproduction, to reach favorable areas for egg laying or rearing of young ones. It helps in distribution most of the animals from plant. This is the most important characteristic, that distributions animals from plants. So locomotion increases the survival chances, okay, and continuation of race, of course. Now, what are the types of locomotion, kinds of locomotions present among the protozoans? So here the four types of mainly four types, that is pseudopodia, flagella, cilia, and pellicular myonemes. They are further classified. Okay, the pseudopodia can be of four types mainly found, discovered till date. So, okay, so Lobopodia. The first one, the four main types of pseudopodia. First one is Lobopodia. These have a broad and blunt ends and are found of both ectoplasm and endoplasm. Example, amoeba, which is multipodal, okay? And entamoeba, which is mono, matlab having a single feet, monopodal, and Lobopodia move by the pressure flow mechanism of water currents, okay? Pilopodia, they have filamentous pseudopodia with tapering ends and are formed of only ectoplasm. Okay, not endoplasm. Okay, the example is Euglophila. Now, reticulopodia or rhizopodia. As the name suggests, reticulo means network. So here you can see in this diagram, there is a reticular, okay, network like filamentous and branch pseudopodia, which is anatomous and forms a network which serves as a trap to capture the prey. Example, foraminiferans. So, ephidium. So just you can go through the glossary and get familiar with the some of the terms. Example, foramines. So marine protozoan with calcareous shell and reticulating pseudopodia. So you can just identify the reticulopodia by this foraminiferans. That is, they have a network-like arrangement, network branch-like arrangement of pseudopodias. Last is exopodia. They are straight and radiating pseudopodia. You can see in this diagram, they are straight and radiating outwards, and they arise from the surface of the body. They arise from the body surface. As you can see here, each exopodium has a central axis filament which is covered by a granular and adhesive cytoplasm. Granular means granules, small granules like, and adhesive means attached. They give an appearance of sun rays and are found in heliosome, that is actinopyres, which are commonly called sun animalcules, because the this shape is like a sun like structure drawn here, so they also can be called as sun animalcules because they resemble the rays of the sun radiating outwards. So like that is that you can also see in the diagram. So they are named as sun-like, or helio means sun-like, and zoans means protozoan. These pseudopodia also show two-way flow of cytoplasm. That is, they can also be from inside to outside and outside to inside. Now, the next we move to the structure of the flagellum. The structure of flagellum. Internally, the structure of flagellum is formed of central in contractile vacuole of axial filaments, or that is also called axoneme. Okay, so surrounded by a thin elastic protoplasmic sheath, except at the tip of axoneme. So except at the tip, the whole thing is covered with axial filament, that is elastic protoplasmic sheath, and it is naked. The axial region is naked. So protoplasmic sheath is a continuous with cell membrane. Axoneme is formed of 11 longitude fibril arrangement in the manner of 9 plus 2 arrangement. 9 fibrils, and that are the doublet and peripheral. Like you can see in the diagram here, they are doublet and they are arranged in 9. They are 9 in total and arrangement in doublet form. So total are 11 like this. While the two peripherals are central and unpaired. So the central ones are unpaired and the ones which are located at the periphery are paired. Now, the two subtubules of each peripheral region, suppose we call them A and B, are collectively form the figure of 8. Okay, so this one is A, this one is B, suppose. So they form a figure of 8, and they're chemically formed of dynein protein, and ATP is enzyme. So what is a dynein protein? It is a motor protein that moves along the microtubules essential for ciliary and flagellar movement. Now, the peripheral proteins are interconnected by inter-doublet link, that is this thing, and BA linkers are chemically formed of next-gen protein. So next-gen protein is a structural protein. As this is a motor protein, this is a structural protein linking adjacent microtubule doublets in a cilia and flagella, enabling bending. So it enables the next-gen protein enables the bending of this thing, and all the 9 protein tubules are arranged in the form of a ring. So on the subfibrils of each peripheral tubule give one pair of small arms. Okay, the one pair of small arms are these small arms, and all the arms are arranged in a clockwise manner like this thing, and Now, all the arms are arranged in a clockwise manner, as we can see, by a common central sheath, and they're interconnected by a double bridge between the peripheral and the central bridge that are 9 accessory fibrils, as we have discussed earlier. Each central tubule is formed of 13 globular tubules, proteins, molecules. So, each flagellum arises from a minute microcytoplasmic granule, that is the basal granule or the basal body, or kinetosome or bleroplast, which supports the flagellum. So what is the bleroplast? A basal body of an organizing center from which a flagellum or coelom arises in a protozoa. So kinetosome generally located near the anterior end of the body, but in trypanosoma, single flagellum originates from kinetosome located near the posterior end of the body, which runs forward along the margin of the undulating membrane and becomes free entirely. Now there are 5 types of flagella, that is steichioniomatic type and pentomatic type and acronematic type, pentaacronematic type, and simple type. The first one, stichonematic type. In this type, protoplasmic sheath of flagellum is produced into single row of short hair-like processes called mastigonemes or flimmers. Example, flagellum of Euglena. Next is pentonematic type. In this, flagella bears 2 or more rows of mastigonemes. Okay, so mastigonemes on the either side of the flagellum. Example, paranema. Acronematic is the absent. The mastigonemes are absent and distal end of the flagellum ends in the terminal flagellum. Okay, example is Eurosoleus. And pentanematic, as we can see that penta means 5. So in this flagellum bears 2 rows of mastigone in each side, also ends to the terminal. So terminal end there are 3 and 2 rows of mastigonemes. So total are 5. Okay, so pentaacronematic. Flagellum of Chelomonas and Cryptomonas are the example. Now the simple type of flagellum is the without mastigonemes and terminal filaments are the simple types. Now let's move to the flagellar locomotion. Flagellar locomotion involves a series of undulating waves passing from base to tip. Okay, so flagellum which pushes the organism in the direction opposite to that of flagellum. This helps in So in this type of locomotion, the flagellar movement is opposite to that of the organism present so that it can help in locomotion. Flagella beat independently and the speed of locomotion varies from 5 micro to 300 micron in flagellates, and the source of energy of flagellar beating is ATP. So let's discuss the swimming mechanism of flagellar locomotion. Flagella is directed obliquely backward towards the backside with a stigma. Okay, so it was founded by Londy in 1943, and flagellum is thrown into a series of spiral undulations at the rate of 12 beats per second. Okay, so next, these extend from base to tip, and the spiral undulations are due to the contraction of contractile fibers of axoneme, consuming energy provided by ATP molecules. The velocity and amplitude of the waves increase towards the tip. As we all know that it originates at the base and it flows towards the tip. So the amplitude and the velocity is maximum at the tip, which confirms that the energy is provided by the flagellum itself and not by the basal body. Flagellar beating involves sliding movement of microtubules on one on other side as in the mycel fibers in humans. Okay, so there is a sliding of a doublet over another doublet. The force of this sliding mechanism is provided by the dynein protein, as we discussed earlier, which acts as the ATPase and hydrolyzes the ATP. This kind of flagellar beating has the threefold effect, that is, it provides the force which pushes the water backwards and forward push. Next, it causes axial rotation at the anterior end on its longitudinal axis. The body undergoes gyration, which is a kind of cyclic rotation. Okay, cyclar or spiral movement, often described in the movement pattern of a certain protozoa, along with the general axis of direction of movement. So, Euglena swims on the principle of aeroplane propeller. So, what is the aeroplane propeller principle? Refers to the spiral rotation movement of flagella in the similar manner of an aircraft propeller, and it spins in a spiral manner along with a straight line, keeping the same body surface facing the general axis of locomotion. So the speed of flagellar locomotion in Euglena is 3.6 mm per minute. So you can see that this is the mechanism of flagellar movement. So the flagella of Euglena is entirely located but provides a forward push to it. So it is called pulsellum or and not traculum, which pulls the body. So it is a kind of push, not a pull. As in Euglena, the direction of spiral beat of flagellum cannot be reversed, so it cannot swim backwards. So it is always swimming in forward direction. Among the choanoflagellates, Proterospongia is with the amoeboid cell in the interior, while the choanocytes at the periphery are present. Periphery means the outer sphere. So act as a connecting link between the protozoan and the sponges. So this is also an important point here to mention that choanoflagellates and Proterospongia are the connecting links in which because the amoeboid cells and the choanocytes both are present. Amoeboid cells are present at the interior region and choanocytes are present in the periphery. So these are the connecting link in the course of evolution. The next thing is cilia. Cilia are also important locomotory organs and they help in locomotion in Paramecium mostly. So they are more in number as compared to pseudopodia and flagella, and they are hair-like, highly vibrating ectoplasmic processes which are organelles of locomotion of most of the ciliate protozoa, except sedentary ciliates like Ephelota and Podophyta. These are generally distributed on whole body surface, cilia of the पेरिस्टोमल रीजन. इंटरनली ईच सीलियम इज सिमिलर टू द फ्लैगेलम. तो अंदर से तो सारे सिमिलर ही होते हैं, बट इट ऑल्सो अराइजेस फ्रॉम द ectoplasmic basal granule, but these do not bear mastogonemes as found in flagellum. Okay, and mastogonemes, they are present at the base in the flagella, so these are not present in cilia. Cilia always beat in a coordination, either synchronously, that is cilia of longitudinal lobes, or metachronously, that is cilia of transverse lobe beating simultaneously. Among to the protozoans, the cilia are faster in their locomotion. Okay, so cilia are much faster as compared to, because they are numerous in number and cover about 2 mm per second. The coordinated beating of body cilia is controlled by intrafacillary or neuromotor system, which is formed of cilia, basal granule, kinetosomes, neuroneme, a fiber arising from the basal granule, that is kinetodesma, a bundle of neuronemes, kinetins and motoneme. Example of these are Paramecium. Okay, the main example of cilia processing organism is Paramecium. So mainly it is discussed the swimming locomotion mechanism in Paramecium, and it is a rapid type of locomotion. Okay, and the speed is 1.5 mm per second. So it covers this distance in 1 second, controlled by rhythmic beating of body cilia in a metachronous or isochronous manner, which is coordinated by a neuromotor system. Energy for ciliary beating is provided by ATP formed during cell respiration inside the mitochondria. It is aided by a streamlined body shape which reduces the resistance offered by water. In metachronous beating, cilia of the transverse manner beat synchronously, while cilia of longitudinal row beat in a characteristic wave manner, which extend anterior posteriorly like the lashing of wheat plants in the strong wind. As you have seen that sometimes wheat plant turn towards a direction in which the wind is flowing strongly, they are bended towards the, like in that manner, the wave manner, which extend anterior posteriorly like the lashing of a wheat plant. Swimming involves three processes, that is a forward push. The first thing is a forward push, so provides by the beating of cilia, which involves sliding movement of microtubules one over the other in the muscle fiber like actin slide over the myosin filament. So each ciliary beat is formed of two strokes. What are the two strokes? Number one is effective stroke or power stroke. During this, cilium becomes stiff due to contraction of axoneme and moves backwards with a rod, like a rod with a force. And recovery stroke is during this, the cilium becomes flexed due to elasticity of the protoplasmic sheet and move forward in a flex position to the original position. So these are the two strokes in a forward push direction, and in the serial beating the power stroke is more powerful than the recovery stroke. Of course it has to be the more powerful one. So this gives a forward push, so that serial beating is like in the row of a boat, like there is a rowing of a boat with the help of a OR. So like that only the ciliary beating is done in the forward push. Now next we move to the amoeboid locomotion. It is a characteristic type of locomotion of all sarcodines. A certain mastigophores, example mastigo amoeba. So certain sporozoan and phagocytes speed of locomotion varies. And it also involves the formation of new pseudopodia in the direction of locomotion. So there involves the formation of pseudopodia in the direction of locomotion over the surface of the streamlining flow of cytoplasm, while at the same time old pseudopodia are withdrawn from the opposite side. So in the side where the locomotion is happening, the pseudopodias are formed, and where there is no locomotion, they are withdrawn. Several theories have been put forward to explain the mechanism formation, that is contraction hydraulic theory, surface tension theory, rolling movement theory, walking movement theory, but the most accepted one is amoeboid locomotion theory, that is change of viscosity theory or soul gel theory. So now let's discuss the soul gel theory. It was provided by Hyman, Pentic, Mast, Reddiff and John. It enjoys a wide acceptance. According to theory, pseudopodia are formed and withdrawn due to cyclic viscosity. Okay, the change in consistency of cytoplasm from soul to gel and gel to soul states. So here is a diagram given to explain the soul gel theory by Mast. This is mentioned as pseudopodium. This is the outer surface of plasma lemma. This is the endoplasm. Here it is, this is the plasma gel, and the inside one, the fluid is plasmosol. Okay, the inside one is soul and the outside one is the gel. This is a uroid because it has wavy structure. Plasma gel. So there is a simultaneous changing of these body fluids, interchange of these body fluids. According to this, cytoplasm of amoeba consists of an outer layer, jelly-like consistency, which surrounds a body fluid. Okay, fluid mass. The former is called a plasma gel and the later is called a plasma soul. So locomotion in this way was explained by four main processes occurring simultaneously. The first one is outermost thin elastic plasma membrane of plasma lemma becomes attached to the substratum. Now but the next thing happens is the local partial liquefaction of the plasma gel at the anterior end, and this causes the central plasmosole under tension. So a tension is built up to flow the forward push, and the force of the plasma gel against this weakened area to produce a bulge, indicating that the beginning of pseudopodium. Here is a bulge and the direction of movement is also in this direction. So in this direction, in this particular direction in which the bulge is happening, there is a movement and also the force. Okay, the exertion of the force is in the bulging side of the movement. So force of plasma gel against this weakened area to produce a bulge, indicating the beginning of pseudopodium. So here the bulging end produces a pseudopodium. As a plasmosole enters the newly formed pseudopodium, it rapidly changes into plasma gel around a periphery, thus forming a gelatinized tube within which the plasma gel continues to flow forward. So in this way, it gives it this a forward push so that the so that pseudopodia can come out of the body and cause a forward movement. Posteriorly, the inner surface of the contractile plasma gel undergoes solation, that is, become fluid. Thus, a consistent flow of plasmosole is maintained from behind forward in the direction of movement. Next thing what happens is the outer tube of the elastic plasma gel contracts and moves from in backward direction while the main bulk of the body travels forward, and the plasma gel thus exerts a squeezing motion from the sides and near of the amoeba, forcing the plasmosole ahead. Thus, locomotion of amoeba is compared to the tube of contractile plasma gel, which is elongated itself by adding into its length at the anterior and shortening of the posterior end by changing into plasmosole. In some sarcodines, like Mastigoamoeba, both the pseudopodia and plasma are present. So, as the organelles of locomotion, the connecting link between the flagellates and the sarcodines. Last thing was the flagella, which we have discussed previously. Now, let's move to the next. Thank you and follow for more.